Turn on the computer. Turn on the instrument. Wait until the checkmark indicator on the front panel of the instrument is flashing before starting the MiControl software. Check that the buffer tank, wash tank, and waste tank are all within the correct volume range. Open the MiControl software. Follow the on-screen instructions to complete the instrument setup. Follow the instructions to load the select chip, syringes, and reagents. Open the select chip package. Handle the chip by the plastic case and be careful not to touch the cover glass. Load the microfluidic chip in the front slot of the stage and press firmly down on the handle until the red spot appears on the side. Attach the tubing to the syringe by screwing it clockwise. Match the color of the syringe with the color of the holder on the syringe pump. Turn the syringe clamp to latch the syringes firmly in place. Align the sample withdraw tubing with the slits and let them hang down above the sample carrier. The black line on the injection tubing, going to the chip, should be aligned with a white arrow. Push down firmly when closing the tubing cover plate. Close the instrument cover. Open the reagent kit. Spin down the fixation reagent, staining reagents, and wash reagents. Place the individual reagents into the reagent carrier by matching the cap number and location number. Place the wash reagents in the remaining empty locations. Remove the caps from all reagents. Align the white line on the reagent carrier with the instrument and slide it up. Twist the module to lock it in place. After loading the chip and reagents, follow the on-screen instructions to initialize the chip. Then go prepare the samples. Mix the sample 3 times by inverting the sample tube. Transfer 2 4 milliliter aliquots of blood into 2 correspondingly labeled 50 milliliter conical centrifuge tubes. Centrifuge 135 microliters of the sorting reagents using a 0.1 micrometer filter cap at 12,000 times gravity for 2 minutes. Transfer 64 microliters of the filtered sorting reagents to each of the 4 milliliter samples. Mix slowly and gently by pipetting 2 times. Incubate at room temperature in the dark for 20 minutes on a rocker with a 60-degree rotation angle at 10 swings per minute. After incubation, remove 2 milliliters from each sample tube and transfer them to new 50 milliliter conical centrifuge tubes, leaving 2 milliliters behind. Add 24 milliliters filtered isoton 2 diluent into each tube. Centrifuge the samples at 800 times gravity for 10 minutes in a swing bucket centrifuge at room temperature with the brake off. Following centrifugation, slowly remove 24 milliliters of supernatant from each tube, being careful not to disturb the bottom blood cell layer. Slowly mix the remaining sample 10 times by pipetting. If needed, mix additional times. Combine the 2 milliliter aliquots by pipetting one back into the original 50 milliliter tube for a total of 2 tubes with 4 milliliters each. The final volume of each tube needs to be the same. Add isoton 2 diluent as needed to balance the volume on a scale. Load the samples onto the MiSelect R system within 30 minutes. Place the prepared samples on the sample carrier. Make sure that the tubing plates are fully inserted into the centrifuge tubes. After loading the samples, follow the on-screen instructions to process the samples. Once the sample processing is complete, you can view the cells. After the experiment is completed, follow the on-screen instructions to remove the select chip, reagents, and samples from the instrument. Remove the select chip, reagents, and samples from the instrument. Dispose of waste according to local regulations. Exit the MyControl software or start a new experiment. Turn off the instrument. Turn off the computer.